Hey class, so here we come back with another important topic, and this is entitled as synapse. And this synapse is actually a structure, and in the structure there would be a mechanism taking place, and the name of the mechanism is synapses. Okay, like if I were to just recall some of the concepts from the previous lecture. So here you see the situation was that we have the receptors in our body and they would be detecting a change in the environment and then they would ultimately convert it into an electrical impulse signal. And that would be conveyed to this neuron that we have, and this was a sensory neuron. From the sensory neuron, the information was going to this relay neuron that we have, and ultimately it would come to this motor neuron that would be taking the message or electrical impulse to the muscles or the glands of the body. But if you were to see this diagram clearly, so the problem here is that this sensory neuron and the relay neuron and the motor neuron, they are not directly connected with one another, but rather than there exists a gap between them. Okay, so we actually have to crack for today is that if there is a gap here, then how come the electrical impulse or the electrical signal that is being conveyed in the electrical communication followed by the body, so this would be transmitted. So this is like the problem as I've said at the start that the neurons are not directly connected, but rather than the gap exists between them, and we have to be thinking of some situation to be considering this. So here let us just try to see that how come the structure of the synapse would look like. But before I proceed with this, what you need to be keeping in your mind is that if I have got the end of the sensory neuron, so this would become presynaptic neuron, and the start of the relay neuron would become postsynaptic neuron. Likewise, if I were to talk the association between the relay and the motor neuron, so the end of the relay neuron would become presynaptic neuron, and the start of the motor neuron would become the postsynaptic neuron, we say. So for the synapse structure that includes that there would be this presynaptic neuron with us. After presynaptic neuron, there would be the postsynaptic neuron with us. So this is the postsynaptic neuron that we have got here. And there exists a gap between them, and this gap is the synaptic gap, or we also call this gap as the synaptic cleft. And the situation here is, if I were to zoom this, so in the presynaptic neuron, we have got this neurotransmitter vesicles, and in the neurotransmitter vesicles, there would be presence of these molecules, and these molecules they are referred to as the neurotransmitter molecules. Now, moving onward, if you were to see this postsynaptic neuron membrane that I have with me, or the neuron I have with me, so here we have these protein receptors. And I believe you would be getting an idea that if I have to carry the electrical impulse from here to here, then these neurotransmitter molecules, by some mean, they should be coming from here to here into the synaptic cleft, and then they should bind with these protein receptors. In that way, the electrical impulse would be transmitted. So if I were to show you a neat diagram of this, so this is the same diagram that I have explained above, but is a neat one. So here, this is the sensory, here we have the relay, and then afterward, we have got this motor neuron, and that motor neuron we have discussed in the concept of the reflex section that this would be escaping through the spinal cord. Now, again, as of before, here we have this presynaptic neuron. Here we have the neurotransmitter vesicles. At the center in them, there is packaging of the neurotransmitter molecules. This is a synaptic gap or the cleft, and then finally, here we have got this postsynaptic neuron, and then there would be the presence of these protein receptors, and these protein receptors, their shape has to be complementary to these neurotransmitter molecules that we have with us. So here we would quickly look on toward the mechanism that how come the electrical impulse would be transmitted from the pre to the postsynaptic neuron. So you see the diagrams for the while, and then I will take you to the explanation that you are required to be writing it down in your exam. So the first step would be that the electrical impulse would arrive in the presynaptic neuron. And when this would arrive in the presynaptic neuron, then as a result of that, if you were to see this, that we have these neurotransmitter vesicles, and these neurotransmitter vesicles, they would compel to move toward the membrane of the presynaptic neuron, and here they would fuse with it. And the time when they fuse with it, so as a result of that, these neurotransmitter molecules that we have, so they are released into this gap in between the pre and the post synaptic neuron, and this was the synaptic cleft or the synaptic gap. So now moving onward, now in the postsynaptic neuron, these protein receptors, they would bind with these neurotransmitter molecules because the shape of these is complementary to each other. And the time when they would bind here to the protein receptor, then ultimately in the postsynaptic neuron, you see that the electrical impulse would be transmitted. And the other idea is that after we have caused our muscle to contract, like once we have compelled the effector to be coming into the play by the muscles, we will allow it to contract, and the gland, we would allow it to be releasing the hormone out of it. So these neurotransmitter molecules, they would be broken down, and after they are broken down or they are removed from these protein receptors, and again they would be reabsorbed back into the presynaptic neurons so that we can again pack them within the neurotransmitter vesicles that we have got. So if I were to take you to the explanation of this mechanism, so you would say that initially an impulse would arrive at the presynaptic neuron. And then the vesicles that we have here, they would move toward the membrane of the presynaptic neuron, and they would release the neurotransmitter molecules here into the synaptic cleft. And then you see that at this point, like if I were to take you a bit back here, so at this point, the concentration of the neurotransmitter molecules is higher, and on the protein receptor, this is lower. So the mechanism by which these neurotransmitter molecule within the synaptic cleft, so this is by diffusion process. So they are diffusing down the synaptic cleft, and then later they would bind to the protein receptors. And then you see here, as if this is written, that these neurotransmitter molecules, and they bind to the receptors, the protein receptor that are available on the postsynaptic neuron, and that would trigger, or that would be making an electrical impulse in the postsynaptic neuron, and then it would be sent to the effectors of your body. Now, here we must also know that what significance do we have for this synapses mechanism? The situation is that this only allows information to be carried in one direction only. This would not be carried in two directions. And the examiner would ask the reason for this. So the situation here is that we only have got the neurotransmitter vesicles having the neurotransmitter molecules only here in the presynaptic neuron and their protein receptors. They're always available, or they are always present on the in the membrane of the postsynaptic neuron. So the information would keep on going only in this one direction that I am showing you at the moment. And the other role that we have in the memory of the synapses is that it would develop the memory in our brain. Like if I were to take you to a situation where you went to some place and over there you're meeting a person for the very first time and he has applied some good perfume on him. So a new synapse would be developed there, and later in your life, if you would come across the same person, so you would have a memory of that person. Now here there are some of the past paper tips that I have to tell you. If the examiner says that what happens when you cut the sensory neuron, so when you cut down the sensory neuron, you would literally would not feel any of the sensation of the pain, and neither you would cause your muscles to contract. And if they say that we are cutting down the motor neuron, or the motor neuron got damaged by some mean or the other, then here you would be feeling the pain because before that the sensory and the relay neuron are working, but you would not be able to cause your muscles to be contracting or the glands to be releasing the hormones out of it. Now there is another important concept that is being asked in the exam. Here they would tell you that there is this normal person and he has got more neurotransmitter vesicles and the neurotransmitter molecules packed with them, and there is a person who is suffering from some disease, and in the disease the number of the neurotransmitter vesicles is less and their neurotransmitter molecules are also less. Then what would be the effect of such thing on the person? So you would say that if the diseased person has less number of the neurotransmitter vesicles, so these neurotransmitter vesicles would move less toward the membrane and they would fuse less with this membrane, and then as a result they would release less of the neurotransmitter molecules into the synaptic gap. And as a result, the binding of the neurotransmitter molecules with the protein receptors would be affected, and as a result, their responses will be very much slow for them. Now again, the situation is that comes in the exam is that how come the anesthetics they work? What do we mean by the anesthetics? That when you are performed some surgery, so the doctors they give you the injection of this anesthesia, and under that you don't feel any of the pain. So how do the anesthetics work? Like either they would block these protein receptors on the postsynaptic neuron membrane, so that means that the neurotransmitter molecules cannot bind with these protein receptors, and ultimately the electrical impulse cannot be further carried with the help of the postsynaptic neuron. Or another way, like after we have caused this neurotransmitter molecule to be binding with the protein receptor, so our muscles they would keep in the contracted state. And if I want my muscle to relax again, then normally in the body there are some enzymes, and these enzymes they break down these neurotransmitter molecules. So these anesthetics, by some way, they block these enzymes up. And when these enzymes they are blocked, then these neurotransmitter molecules they would keep binded with the protein molecules, and ultimately the new pain perception cannot be sensed, and neither it can be going to your muscles to be causing them to contract. So I hope you would be clear with the concept of this synapses and synapse that we have got working for our nervous coordination and the response phenomenon. So see you in the next class.